Welcome back, everyone, to the deep dive. We're going to be diving deep today into the world of biotechnology. You've given us some really cool textbook excerpts, and it looks like we're going to be talking about like everything from the origins of like genetic engineering all the way to like test tube babies.
Oh, yeah.
So
all the exciting things.
This should be pretty wild.
It is a fascinating field.
So I guess to kick things off, like what is biotechnology?
Biotechnology, it's interesting. A lot of people think it's a very new field.
Oh, really?
But it's actually been around for centuries in its traditional forms.
Oh, wow.
So if you think about a lot of the foods that we eat every day, like bread and yogurt and cheese, those are all created through traditional biotechnology processes using living organisms to make delicious and useful things.
So we've been manipulating the biological world for a long time.
We have.
But it seems like today it's going far beyond bread and cheese, right?
Absolutely. Today, biotechnology is used for things like developing new medicines, creating vaccines, and even correcting genetic defects. And this revolution in healthcare and agriculture is all thanks to genetically modified organisms.
Wow. So manipulating like these tiny organisms can have a huge impact on...
Huge impact on our lives.
Yeah.
But how did we get to this point?
So believe it or not, the roots of modern biotechnology actually go back to the 17th century. So the philosopher René Descartes had this idea that science should be used to improve human life and, make us masters and possessors of nature. It's a very human-centered view of science.
So it wasn't just about, like, understanding the world.
Yeah. It was about using that knowledge.
Yeah, using it to our advantage.
To improve our lives.
Yeah, that's really fascinating.
It is. Yeah. And if you think about how, physics and chemistry, those led to all those amazing inventions during the Industrial Revolution, biotechnology followed a similar path. Emerging from biology to tackle challenges in food production and medicine. Okay, so we have this new field of biotechnology, but who are some of the key players in like making this all happen?
That's a great question. I mean, obviously it was a collaborative effort with many brilliant minds.
Yeah.
But one name that stands out is Herbert Boyer, a scientist whose journey is anything but ordinary.
I like that.
Yeah.
Tell me about Boyer.
Yeah, so he grew up in a small Pennsylvania coal mining town, where most of the young men just followed a predictable path working in the mines or on the railroads.
Yeah.
But Boyer had a different vision.
Yeah.
He really pursued his education. He ended up earning a PhD from the University of Pittsburgh, spending time at Yale for further studies, and ended up as a professor at the University of California, San Francisco.
He definitely did not take the easy route.
He did not.
So what was it about?
So in 1969, Boyer was studying E. coli bacteria, and specifically the enzymes that they produce, called restriction enzymes. Yeah. And his groundbreaking discovery was that these enzymes could cut DNA strands at very specific points, leaving what scientists call sticky ends.
Sticky ends? I've never heard of that before.
Have you ever cut paper with scissors that, like, leaves the edges a bit jagged?
Oh.
Allowing them to overlap?
Yes.
So those overlapping edges would be sticky and could easily be reattached.
Okay.
It's a similar concept with DNA. These sticky ends allow scientists to cut and paste DNA segments with incredible precision.
So they discovered the molecular...
It's like discovering the molecular scissors and glue.
Scissors and glue.
For genetic engineering.
Wow.
That's pretty powerful.
Yeah, I could see why that would be like a revolutionary.
Revolutionary.
Yeah. But I'm guessing he wasn't working in isolation.
You're absolutely right. So Stanley Cohen, a scientist at Stanford, he was studying these things called plasmids, which are like tiny independent loops of DNA that can replicate within bacteria. And Cohen figured out a way to remove these plasmids and put them back into bacterial cells.
Okay, so we have...
So you have Boyer with his DNA scissors and Cohen with his method of moving DNA around.
Yeah.
And their paths collided in Hawaii, of all places, during a brainstorming session. And they realized the potential of combining their expertise. So Boyer's DNA splicing technique with Cohen's plasmid delivery system.
Wait, so they were like thinking about inserting specific pieces of DNA into bacteria and then
Yes.
having them create like proteins and stuff.
That's exactly it. That was the birth of recombinant DNA technology, the foundation of modern biotechnology.
Wow, that's crazy.
It is.
But hold on, I need to back up for a second.
Yeah.
What are the core principles of genetic engineering? Like, how does it actually...
So there are two key techniques. First, we have genetic engineering itself,
Yeah.
where you manipulate these DNA and RNA sequences and then insert those altered sequences into a host organism to change its traits.
So that's where the like molecular
Molecular cutting and pasting comes in.
comes in?
Yeah.
Okay, but then...
But then you need a place for those organisms to do their work.
Right.
That's where the second core technique comes in, bioprocess engineering.
Okay.
And it's all about creating sterile, controlled environments, sometimes massive vats, where these genetically modified organisms can be grown on a large scale to produce things like antibiotics or vaccines.
So that's how we get enough of it to
Exactly.
be useful.
Yeah.
That makes sense. Okay, so before we dive into all the
Right.
like nitty-gritty details,
Uh,
I think we need to understand like why this works in the first place.
right.
Like
Okay.
why are we able to do this?
Let's step back and think about something we're all familiar with. Sexual reproduction.
Okay.
You know, it creates these new combinations of genes,
Mm-hmm.
leading to diversity
Right.
within species.
Uh-huh.
And this mixing of genes is incredibly beneficial
Yeah, diversity is good
for individuals.
for everyone.
The survival of whole populations. Now, think about how farmers have been trying to improve crops and livestock for centuries.
Yeah.
They do this by crossbreeding different varieties to get the best traits, right?
Yeah, but like with that method,
But you end
Shit.
up with a mix of both desirable and undesirable traits.
yeah.
Because you're transferring large chunks of DNA. It's like throwing a whole bag of seeds in the ground, hoping to get the best plants.
Okay, I see what you're saying.
Yeah,
So with genetic engineering, scientists have like way more control over
exactly.
what genes are transferred, right?
Precisely. Using techniques like recombinant DNA technology, scientists can pick and choose the specific genes they want to transfer. They can get the juiciest tomato genes without all the random extras,
Okay.
you know?
That's awesome.
Yeah.
So can we go back to like
Yeah.
that idea of inserting the DNA into a different organism?
Sure.
What actually happens to that piece of DNA once it's in there?
That's a great question. Imagine we take a piece of DNA from one organism and just insert it into another. What do you think will happen to that DNA?
Well, I'm guessing it has to somehow integrate
You have
with the host
Yeah.
DNA to have an effect, right?
Exactly.
Yeah.
It needs to become part of the host's genome.
Okay.
Its genetic blueprint, because if it doesn't integrate that foreign DNA,
Mhm.
it's not going to be able to replicate and pass on its information.
So it needs to be a part of the host's DNA, but how does that happen?
Does that happen?
Well, there are these specific DNA sequences called origins of replication,
Okay.
and these sequences act like starting points for the replication process. For a piece of foreign DNA to be copied, it needs to be linked to one of these origins. It's like attaching a train car to a locomotive. Without that connection, it's not going anywhere.
Oh, that's a great analogy.
Yeah.
So like if you're trying to clone something,
Exactly.
you're making a lot of copies.
You need to link it
You need to link it to...
to that origin of replication.
Yeah, okay. Now let's go back in time a little bit and talk about the first successful creation of an artificial recombinant DNA molecule.
Okay.
This was a pivotal moment in the history of biotechnology. This was in 1972
Okay.
with, our dynamic duo Stanley Cohen and Herbert Boyer.
Okay.
They created a recombinant DNA molecule by linking a gene that provides antibiotic resistance to a native plasmid from Salmonella typhimurium bacteria.
Okay, wait, what's a plasmid again?
So remember, it's like a tiny
Oh, yeah.
circular piece of DNA
Yeah.
that can exist and replicate independently inside a bacterium.
Okay.
In this case, the plasmid acted as a vector.
Okay.
It's a vehicle for delivering the antibiotic resistance gene into a host organism,
So like a Trojan horse,
like a Trojan horse,
sneaking the gene in.
sneaking that gene into the host cell.
Oh, I like that.
And to create this recombinant DNA molecule, they used an enzyme called DNA ligase,
Okay.
which acts as molecular glue, joining the cut ends of DNA fragments together.
It's like so elegant how,
Very elegant.
and they're able to do this.
Yes.
Yes.
It's amazing what nature has
Like,
provided us.
So they created this.
So they transferred this recombinant DNA into E. coli bacteria,
Okay.
which is a close relative of Salmonella.
Okay.
The E. coli served as the host, providing the machinery for the recombinant DNA to replicate and create multiple copies of that antibiotic resistance gene.
Hold on.
Yeah,
How did they get the recombinant DNA inside the E. coli in the first place?
good question. So remember how we talked about plasmids being like delivery vehicles?
Yes.
Well, they can naturally transfer DNA between bacteria. In this case, the plasmid carried the recombinant DNA, including the antibiotic resistance gene, into the unsuspecting E. coli.
So it really was like that Trojan horse sneaking the gene in.
Sneaking it in.
I like that analogy.
Yeah, and this marked the first successful cloning of a gene,
Oh, wow.
a major turning point in biotechnology.
That's crazy. Okay, so we've talked about that first one, but I'm sure it's become
Oh, it's much more refined now.
much more
Yeah.
refined over time.
Yeah.
Can you break down the steps involved in...
So we can boil it down to three main steps.
Hmm.
First, you need to identify the DNA sequence that contains the gene you want, the trait you want to introduce to your host organism. It's like finding the instruction manual for building a specific feature.
So it's like finding the genetic code for that.
Exactly.
Okay.
Then you have to introduce this identified DNA into the host organism. Scientists use all sorts of different techniques for this, including plasmids, microinjection, or even gene guns.
Oh, wow. So many different ways to get it
They are.
in there.
Yeah.
And then finally,
Finally, you need to make sure that introduced DNA is stable in its new home so it becomes a permanent part of the host's genetic makeup and can be passed on to future generations.
so it can't just get in there. It has to like
And to stay there.
stay there,
Yes,
like a house guest who never leaves.
exactly.
Okay, so I think I'm starting to get like the big picture.
Yeah.
But let's talk about the tools
Yeah.
in the toolbox, like how...
What's in the molecular toolbox?
Yeah, what's in there?
So we've already talked about restriction enzymes, those molecular scissors for cutting DNA. We also have polymerase enzymes, which are essential for replicating DNA. And then we have ligases, which act as molecular glue to join DNA fragments.
Okay, so those are like
Those are the ones that...
for the DNA itself.
Yeah.
What else?
You also need vectors, which act like delivery trucks to transport the DNA into the host organism. We've talked about plasmids and bacteriophages. And of course, you need a host organism
Okay, makes sense.
to actually be the recipient.
So let's start with those molecular scissors.
Okay.
The restriction enzymes.
So restriction enzymes, they were actually first discovered in 1963. And it turns out that they actually play a role in the bacteria's own natural defense system,
Really?
protecting them from viruses.
So it's part of their immune system.
It is.
That's so cool.
Yeah, there are two types of enzymes involved.
Right.
One adds a special chemical tag to the bacteria's own DNA, so it marks it as self. The second type, called restriction endonucleases, act as the molecular scissors,
Okay.
cutting up any DNA that doesn't have that self tag.
So it's like protecting its own DNA.
And it is. It's a very clever system.
Other bacteria.
The first restriction endonuclease ever isolated was called Hindi, and this enzyme always cuts DNA at the same specific 6-base pair sequence. It's like a key that only fits one specific lock.
Oh, that's a good
Mm.
analogy.
Yeah.
But are all restriction enzymes the same?
Not at all. Over 900 restriction enzymes have been discovered
Wow.
in over 230 different strains of bacteria, and each one recognizes a unique DNA sequence.
So many.
It's like having a giant toolbox full of different scissors, each designed for a specific cutting job.
Okay, but how do they
Well, they have a naming system.
keep track of all that?
Yeah, so the first letter comes from the genus of the bacteria, then the next two from the species, followed by the strain designation, and then Roman numerals to indicate the order in which the enzymes were isolated from that strain.
Okay, so E. coli, I think I saw that one.
Exactly. So E. coli comes from Escherichia coli. The R denotes the particular strain of E. coli, and the I means it was the first restriction enzyme isolated from that strain.
Okay, that makes sense.
Yeah.
But how do these restriction enzymes work? Like, do they just like chop up the DNA randomly?
Not at all. They are very precise. They belong to a group of enzymes called nucleases. There are two types of nucleases, echonucleases, which remove nucleotides from the ends of DNA strands, and then endonucleases, which cut within a DNA strand,
So the restriction enzymes we're talking about
are endonucleases.
endonucleases.
They scan along a DNA molecule, and when they find their specific recognition sequence, they bind to the DNA and cut both strands of the double helix at very precise points. It's like they have a built-in GPS that guides them to their target.
Wow, it's like
It's very
so precise.
precise.
Okay.
And these recognition sequences,
Is there anything special about those?
They're often palindromic sequences,
Oh,
which
right.
means they read the same backward and forward, like the word race car.
Oh, okay.
So a palindromic sequence in DNA would be a sequence of base pairs that reads the same on both strands
Mm. Okay,
when
that's
you read them in opposite directions.
neat.
So where exactly do these enzymes make the cut within this palindromic sequence?
Yeah.
They cut a little bit away from the center of the palindrome between the same two bases on opposite strands,
Ah,
and that's how they create those sticky ends.
sticky ends are back.
They are back.
But why are they called that?
They have overhanging single-stranded portions that are complementary to each other.
Okay.
And those complementary ends can easily form bonds and rejoin. It's like puzzle pieces fitting together or molecular Velcro.
Oh, that's why they're so important for creating recombinant DNA. You can cut it from different sources
Exactly.
and then stick them together.
Yeah.
Oh, that's awesome.
It is, but
Yeah.
once you have
So once you have,
it all cut up, how do you
you
actually,
separate those fragments.
yeah, how do you separate them and find the one you need?
That's where a technique called gel electrophoresis comes in. It's a really clever way to separate DNA fragments based on their size.
Oh yeah, gel electrophoresis.
You remember that?
Yeah, it involved electricity, right?
Yeah, it does. So picture a slab of gelatin. That's essentially what the gel is.
Okay.
We load the DNA fragments into little wells at one end of the gel, and then we apply an electric current across it. Since DNA has a negative charge, those fragments will be drawn toward the positive electrode.
So the smaller ones will move faster.
The smaller DNA fragments will navigate that gel more easily
Yeah.
and travel further than the larger fragments.
Okay, so you end up with a pattern of bands, and each band represents...
DNA fragments.
It's a certain
size.
Okay.
To visualize these bands, we use a special dye that makes the DNA glow under UV light.
Oh, wow.
Yeah.
So now you can see it. And then once you've spotted the band that contains your DNA fragment with your gene of interest,
Okay.
he has to isolate it from the gel.
Okay.
It's like carefully extracting a precious gem from the surrounding rock.
And that.
That process is called elution.
Elution.
Yeah, gel electrophoresis is a really powerful tool for separating and analyzing these DNA fragments, and it's a crucial step in many genetic engineering experiments.
Okay, so now you've got it all isolated. What happens next?
Now we need to introduce that isolated DNA fragment into a vector. Remember, the vector acts as the delivery vehicle
Right.
to carry the DNA into the host organism.
And that was like the plasmids and stuff, right?
Yeah, and we talked about plasmids. But there's another commonly used vector called a bacteriophage, which is a type of virus that infects bacteria.
Wait, you're using viruses?
We are.
Okay, this.
Yeah, and there are good reasons for using bacteriophages as vectors. For one, they can accommodate much larger pieces of DNA than plasmids can,
Okay.
and they also have very high copy numbers.
What's a high copy number?
It refers to the number of copies of the vector that exists within each host cell. Some plasmids only have 1 or 2 copies per cell, but bacteriophages can have hundreds or even thousands of copies per cell.
So that means you can produce a lot more of the...
You can
What
produce
the
a lot more of your gene of interest.
That makes sense.
So this becomes really important when you are trying to produce a protein or another valuable product from that gene.
Okay, so we have plasmids and bacteriophages. But what makes a good vector?
That's a great question. So a good cloning vector needs to have several key features. First of all, it needs to have an origin of replication, or ORI for short. This is a specific DNA sequence where replication begins.
Okay.
And if you link your foreign DNA to the ORI, that ensures it'll get replicated.
Oh, so it's like the...
It's like attaching your important document to the only copy machine in the office.
I like that.
Yeah.
What else?
A good vector also needs a selectable marker.
What's that?
So this is a gene that helps you easily identify which host cells have taken up the recombinant DNA.
Oh, okay.
One common type is an antibiotic resistance gene.
Oh, okay.
So the vector carries a gene that makes bacteria resistant to a specific antibiotic. If you grow these bacteria on a special dish with that antibiotic, only the bacteria that took up the vector, and therefore the resistance gene, will survive.
So it's like it only lets in the ones with like the...
Exactly. It's like a bouncer at a club, only letting in the bacteria with the right ID.
Yeah,
You know?
I like that.
Yeah.
Okay, so
Another crucial feature
what else?
is cloning sites.
Cloning sites.
Yeah, so these are unique restriction enzyme recognition sites.
Okay.
You know, those special DNA sequences we talked about earlier. But these are within the vector's own DNA sequence. These sites make it much easier to insert that foreign DNA into the vector.
So it's like a,
It's like designated spots where you can insert your DNA fragment.
yeah, a little spot for it.
Yeah,
Yeah.
exactly.
Okay, that makes sense.
And ideally, the best vectors only have a few of these cloning sites, making the process simpler and more efficient.
Okay.
Now, we've talked about vectors for bacteria,
Yeah.
but what about for plants and animals?
Oh, no.
There are actually different types of vectors for those as well.
Really? Are they using I don't know, are they using?
So scientists have figured out how to use modified pathogens, like certain bacteria and viruses, as vectors to deliver genes into plants and animals.
Pathogens.
I know it sounds a little scary,
Yeah.
but remember that these pathogens have been disarmed,
Oh.
meaning they can no longer cause disease.
Okay.
A common vector for plants is a modified version of a bacterium called Agrobacterium tumefaciens.
Yeah, that one, doesn't it normally cause
It causes tumors
tumors in plants?
in plants in its natural state. It infects plants and causes these tumors to grow. But scientists have figured out how to remove those tumor-inducing genes from the bacterium's DNA
Oh. Yeah.
and replace them with beneficial genes.
So they're hijacking its ability to insert DNA,
They are hijacking it,
but for good.
but for good.
I like it.
Yeah.
That's really cool.
And then for animals, scientists often use modified retroviruses as vectors.
Retroviruses, like HIV.
Yes, exactly. Retroviruses have this incredible ability to integrate their genetic material into the host cell's DNA.
Right.
So scientists have learned how to harness this ability
That's crazy.
by modifying those retroviruses to carry beneficial genes.
Wow, it's like turning a villain
Turning
into a superhero.
into a superhero.
I like that.
Yeah.
Okay, so you have your gene tucked away in the vector.
What's the next step?
Yeah, what happens next?
Well, we need to get that vector into your host cells, and that process is called transformation.
Transformation.
Yeah.
How do you get DNA, which is a pretty big molecule, to cross the cell membrane? Is that hard to do?
It can be. Cell membranes are very picky about what they let in and out.
Right.
So to get the DNA inside, we have to make the host cells competent to take up the DNA.
Competent. So you have to convince them to let it in.
You could say that
Uh-huh.
one way to make bacteria competent is to treat them with calcium ions.
Okay.
This makes their cell walls more permeable, allowing that DNA to sneak in.
So you're creating little openings
Little temporary openings.
for the DNA to get through.
Exactly.
No.
Once the cells are competent, we can use a technique called heat shock
Heat shock.
to actually introduce that DNA.
What is that?
It's not as dramatic as it sounds.
Okay.
So we incubate the competent cells with the DNA on ice, then briefly expose them to a higher temperature, typically 42 degrees Celsius,
Oh.
and then put them back on ice.
It's like a hot and cold spa day
It is.
for the cells.
It is.
Why does that?
So these rapid temperature changes create a thermal gradient across the cell membrane, and this helps to push the DNA into the cells
So you're using temperature
as a tool.
as a tool.
Yeah. And there are other methods for introducing DNA into cells as well. For animal cells, a technique called microinjection is often used. This involves injecting the DNA directly into the cell's nucleus.
Wow. So it's like
Like a tiny little needle.
tiny surgery.
It's microsurgery for cells.
I love that.
Yeah, and for plant cells, there's a technique called biolistics, which is also known as the gene gun method.
Gene gun.
Gene gun.
What is that?
It sounds very futuristic.
Yeah.
Essentially, tiny particles of gold or tungsten are coated with the DNA.
Okay.
And then these particles are shot into the plant cells using a special device.
So you're just bombarding it with DNA.
Essentially.
That's so cool. And then we also talked about using those disarmed
We did.
pathogens as vectors too, right?
Yeah, we talked about how modified versions of Agrobacterium can be used to deliver genes into plants.
Right.
And modified retroviruses can be used for animals. Both of those are really powerful tools.
Okay.
In the genetic engineer's arsenal,
There's a lot.
there are a lot of steps. But we can summarize the key steps of recombinant DNA technology.
Okay. Yes, please.
First, you isolate the DNA that contains your target gene. Then you cut that DNA and your vector with the same restriction enzyme,
Okay.
creating those matching sticky ends. It's like preparing the edges of two pieces of fabric so they can be sewn together perfectly.
Okay.
Then you use DNA ligase to join the gene of interest to the vector. That's like stitching those two pieces of fabric together,
Okay.
creating your new recombinant DNA molecule.
Okay.
And then you introduce that recombinant DNA into your host organism using one of the transformation techniques we just discussed, effectively delivering it to the cells where it can be put to work.
Okay. That's a lot.
It's a multi-step process.
It is.
but it's very elegant.
But let's break it down even further. What are the steps in isolating the DNA from the source organism?
So first, you have to crack open those cells and release the DNA, sort of like cracking open a nut to get to the good stuff inside. We do this using enzymes that can break down cell walls and membranes,
So you're breaking down those
the barriers.
barriers.
And depending on the type of cell, you use different enzymes. So for bacteria, we often use an enzyme called lysozyme to break down their cell walls. For plant cells, we use cellulase to degrade those cellulose cell walls. And for fungi, we use chitinase to break down their chitinous cell walls.
So it's like using the right tool
The right tool for the job.
for each cell.
Exactly.
Okay.
Once you have access to that cell's interior, you need to purify that DNA. That means removing other cellular components, such as RNA and proteins and lipids. You can imagine it's like separating all the ingredients in a cake batter, and you're left with just the flour,
Okay, so you're getting it all pure
getting it pure.
and clean.
And we use specific enzymes to remove the RNA and proteins. Other molecules are removed using different treatments. And finally, that purified DNA is precipitated using chilled ethanol.
Okay.
This causes the DNA to come out of the solution and form a visible mass.
Yep.
You can actually see it as these fine thread-like strands.
Okay, so now you have the pure DNA.
You have the pure DNA.
Ready for the next step.
Ready for the next step.
What is that?
We need to cut that DNA with a restriction enzyme
Okay.
to isolate the specific fragment that contains our gene of interest.
Okay.
Back to those molecular scissors.
Time to do some DNA surgery.
Exactly. And to make sure that restriction enzyme is working correctly, we use gel electrophoresis to actually check the size of those DNA fragments.
Oh, okay.
Remember how we talked about the DNA migrating through the gel based on size?
Yes.
Well, that's how we can see if the restriction enzyme has cut the DNA at the right location.
So it's like a quality control.
It's a quality control step
Yeah.
to make sure you have the right piece for your puzzle.
Okay.
Once we confirm we have the right piece, we can move on to ligation.
Ligation. That's where you join them.
That's where you join the DNA fragment to the vector.
Okay.
And this is where DNA ligase, our molecular glue, comes into play. It joins those sticky ends of the DNA fragment and the vector, creating that recombinant DNA molecule.
So it's like the thread that's holding the two pieces of fabric together.
Exactly.
Okay, but sometimes you need to make a lot of copies of the DNA fragment.
You do.
Right.
If you're starting with a very tiny amount of DNA,
Okay.
or if your experiment requires many copies of that fragment, you need to amplify it.
Okay.
Think of it like photocopying an important document many times.
So how do you amplify DNA? Is there like a special technique?
We use a technique called PCR, which stands for polymerase chain reaction.
Oh, yeah, PCR. I've heard of that.
You probably have. It's used in all sorts of fields, from
Yeah.
crime scene investigation to medical diagnostics.
It seems like a very powerful technique.
It is. PCR lets us create millions of copies of a specific DNA sequence, even if we only start with a tiny amount of template DNA.
That's crazy. How does it even work?
It involves another one of our special enzymes, DNA polymerase.
Oh, okay.
It's like the molecular copy machine
Okay.
that replicates the DNA sequence.
And what are those primers I read about?
Primers are short, synthetic pieces of DNA,
Okay.
and they're designed to be complementary to the ends of that DNA sequence that you want to copy.
Okay.
They act like starting points for the DNA polymerase.
Mm.
They tell it where to begin copying.
Okay, so they're like,
Start here. This is the important
oh,
part.
this is where we
This is it.
start. Okay.
And here's the really amazing part.
Right.
After each cycle of PCR, the number of DNA copies doubles.
Okay.
So if you start with one copy,
Yeah.
you'll have 2 after 1 cycle, 4 after 2 cycles, and so on.
So it's exponential.
It's an exponential explosion of DNA.
That's so cool.
So you can go from a single copy to millions of copies in a relatively short amount of time.
Wow.
That's why PCR has become such an indispensable tool in molecular biology.
That's incredible.
Also, now you have your amplified DNA fragment
Okay.
ready to be inserted into your vector
Right.
and introduced into that host cell.
Okay, so we're getting close
We're getting close.
to the finish line here.
Now, remember we talked about those antibiotic resistance genes?
Yeah.
That can be used as selectable markers
Right.
to identify those host cells that have successfully taken up the recombinant DNA.
Right. Yeah, so you grow it on the Petri dish. Only the ones with the...
Only the ones that survive have that resistance gene.
Yeah.
But sometimes we need to be even more specific.
Really?
We need to distinguish between the cells that have taken up just the vector
Okay.
versus the cells that have taken up the recombinant DNA, meaning the vector with the inserted gene.
Why is that so important?
Because not every cell that takes up that vector will successfully incorporate the DNA insert.
Oh, okay.
Some might take up the vector without the insert, or the insert might not integrate correctly. We need to weed out those false positives,
Yeah.
you know.
So how do you tell the difference?
One way is to use a technique called blue-white screening.
Blue-white screening.
It involves a special type of plasmid vector that contains a gene for an enzyme called beta-galactosidase.
Beta-galactosidase.
I know, it's a mouthful.
Ah.
But it's actually a pretty simple concept. This enzyme breaks down a sugar called lactose. And when it does that, it produces a blue pigment. So if you grow bacteria that contain this plasmid on a special petri dish with lactose,
Okay.
the colonies will turn blue.
Oh, okay.
Now here's the clever part. The cloning site for that foreign DNA is located within the beta-galactosidase gene.
Wait, so if you insert your DNA into that, you're breaking the gene.
You are.
Okay.
If that gene is broken, the enzyme can't be produced and the colonies won't turn blue.
Oh.
They'll stay white.
So it's a color test.
That's a visual test.
Okay.
Blue colonies mean the bacteria have the plasmid without the insert.
Okay.
And white colonies mean they have the recombinant plasmid with the insert.
I like that.
Yeah, it's a simple but effective way to pick out those colonies.
Okay.
So we've successfully transformed our host cells with the recombinant DNA.
Yeah, what happens next?
Now we need to get those cells to produce that protein
Okay.
that's encoded by our gene of interest.
So it's like you've delivered the blueprints,
You have.
but now you have to make sure
You have to make sure
they actually build the house.
that they actually use them to build the house.
Yeah, okay.
This is called protein expression,
Okay.
and it can be influenced by a variety of factors.
So it's like creating the ideal environment for a plant to grow,
Exactly.
like giving it the right amount of water and sunlight.
Yeah. So what kind of conditions are we talking about for protein expression?
Yeah.
Well, it depends on the protein and the host cell, but things like the promoter sequence that controls gene expression, the growth conditions for the cells.
So there's not like one magic recipe
There is.
that works for everything.
No, it's about fine-tuning it for each situation.
Okay.
And when a protein is produced by a gene that's been expressed in a different organism, we call it a recombinant protein.
Recombinant protein, so that's like the goal of all of this,
That is the goal.
is to make these recombinant proteins.
And why is it so important to produce these recombinant proteins on a large scale?
Yeah, why?
Well, if you're developing a new drug or a therapy,
Yeah.
you need a significant amount of that protein.
Right.
To conduct those experiments, run clinical trials.
It's not just going to be made in like a tiny little...
No, it's like baking a cake for a huge party.
Yeah.
You need a much bigger oven
That makes sense.
and a lot more ingredients.
So how do they scale it up?
We use bioreactors.
Bioreactors, what are those?
Essentially, they're large vessels
Okay.
that are designed to grow cells on a large scale to produce that protein of interest.
So it's like a giant house for all the cells.
It is. And the conditions inside that bioreactor are carefully controlled to optimize the cell growth and protein production.
Okay.
We're talking about things like temperature, pH, nutrient levels, and even the amount of oxygen available.
Like a luxury spa
It's a spa for cells.
for the cells.
Yeah, to keep them happy and productive.
So what kind of bioreactors are there?
One of the most common types is called a stirred tank bioreactor.
Stirred tanks. So I'm imagining like a giant tank with a propeller.
You're not far off. The stirring ensures that the cells and nutrients are evenly distributed throughout the vessel. Most stirred tank bioreactors also have a system for bubbling air or oxygen into that culture,
Okay.
providing those essential ingredients for the cells to grow.
So it's constantly mixing it,
It's mixing it up.
giving it air like a protein smoothie maker.
Yeah, and they're also equipped with all sorts of monitoring and control systems
Okay.
to make sure those conditions are just right.
So it's like it has its own little like thermometers and...
It does. It has sensors for temperature, pH, oxygen levels.
Wow. That's...
And the system can automatically adjust these parameters as needed.
So it's like a little robot caretaker for the cells.
That's a great way to put it. Bioreactors are really impressive feats of engineering.
Okay, so now the bioreactor is making tons of protein.
Tons of it.
What happens next?
The next step is called downstream processing.
Okay.
It involves separating and purifying that protein of interest from all the other stuff that's in the bioreactor.
So it's like panning for gold. You're trying to find
You are
the nuggets?
trying to find the good stuff.
Yeah.
And depending on the protein, downstream processing can be a very complex multi-step process involving things like filtration, centrifugation, chromatography, precipitation.
Wow, that's a lot.
Yeah, it's like using a series of filters to purify water.
Yeah.
You're removing all those impurities.
Okay.
Once we have that purified protein, It needs to be formulated with preservatives,
Okay.
other ingredients, to ensure its stability and make it suitable for its intended use.
So it's not just about getting it.
No, you've got to make it a finished product.
You've got to make it into something you can use.
And for many applications, like developing new drugs, the final product has to undergo rigorous quality control testing
Oh, of course.
to ensure safety and effectiveness.
That makes sense.
It's very similar to the testing process that all new medications go through.
Right.
So we've gone through this entire process, from isolating the DNA to producing this purified protein. It's a fascinating journey.
It is.
But it's important to remember that this technology has far-reaching implications
Okay.
beyond just producing proteins. It can be used to study how genes work, develop new diagnostic tools, and even create organisms with specific traits, like making crops that are resistant to pests or diseases.
So the applications are really broad.
Okay, so let's dive into some of those specific examples.
Okay.
You mentioned that the first recombinant DNA molecule used a gene for antibiotic resistance.
Oh, yeah. Why was that?
Antibiotic resistance genes are often used as selectable markers in recombinant DNA technology.
Okay.
Remember how we talked about using these genes to identify those host cells that have successfully taken up the recombinant DNA?
Yeah, because they're the only ones that survive on the Petri dish.
Exactly.
Okay, so what are some other applications of this technology?
One really exciting area is gene therapy.
Gene therapy. Yeah, that sounds futuristic.
It does.
What is it?
Gene therapy is a really promising approach to treating diseases that are caused by genetic defects. It involves introducing a functional copy of a gene into a patient's cells to correct that problem.
So you're giving the cells the right instructions.
The right instructions.
Okay.
One common approach is to use a modified retrovirus as a vector.
Ah, so they're back.
Back again
Because they can insert their DNA into the
into the host gene.
uptome, right?
So we can modify that retrovirus to carry the therapeutic gene, use it as a delivery vehicle. Hopefully that gene will integrate into the patient's genome, start producing that missing or defective protein, and correct that genetic defect.
Wow, that's like rewriting their
Rewriting their genetic code.
genetic code to fix it. That's incredible.
It's really powerful, but it's still a relatively new field.
Right.
There are many challenges to overcome,
I can imagine.
like making sure the therapeutic gene gets to the right cells, integrates properly, and doesn't cause any unwanted side effects.
Okay, so what about other applications? We've talked about gene therapies or anything else.
Another exciting area is the development of genetically modified crops, also known as GMOs.
Oh, GMOs, I've heard.
You've probably heard a lot about them.
Yeah, a lot of debate.
A lot of debate.
About GMOs, what are they?
So GMOs are crops that have been genetically modified
Okay.
using recombinant DNA technology
Okay.
to give them desirable traits
Okay.
that would be difficult or impossible to achieve through traditional breeding methods.
So you're like giving them
A genetic boost.
the genetic boost.
For instance, scientists have developed crops that are resistant to pests, herbicides, or diseases.
Okay.
And this can help increase crop yields, reduce our reliance on pesticides,
Which is a good thing
which is good.
for the environment.
Yes.
So why all the controversy?
Well, it's important to carefully consider both the potential risks and benefits of GMOs
Okay.
before we widely adopt them. There are concerns about unintended environmental consequences, like
Right.
the development of herbicide-resistant weeds or the transfer of those modified genes to wild populations.
Okay, so there's a lot to consider.
There is.
Both sides of that.
It's a very complex issue with pros and cons.
Okay, so GMOs, gene therapy, anything else?
Another area is the field of diagnostics.
Okay.
Remember PCR?
Yeah.
It's being used to develop really sensitive diagnostic tests for a wide range of conditions.
So not just treating diseases,
No.
but figuring out if people have them.
Early detection is crucial,
Yeah.
especially for serious conditions like cancer.
Okay, that makes sense. So let's talk more about some of those other tools.
Okay.
We talked about restriction enzymes.
Yeah.
What about polymerases and ligases?
So polymerases are the copy machines of the molecular world.
Okay.
They copy DNA sequences. They're essential in DNA replication and PCR.
So they're like the scribes,
That's
like
great.
carefully copying the genetic code.
And then DNA ligases.
Yeah.
Those are the master joiners. They connect those DNA fragments together,
Okay, so they're kind of like the tailors,
like tailors.
stitching it all together.
Restriction enzymes cut, polymerases copy, ligases join.
Yeah, it's like a construction crew.
A molecular construction crew.
Yeah.
And there are many different types of polymerases and ligases,
Okay.
each with their own properties and applications.
It's crazy that nature has provided us with all
I know, it's
those
remarkable.
tools to do this.
And scientists have learned to harness them
Yeah.
to develop these powerful technologies.
Okay, so we talked about enzymes. Are there more vectors besides plasmids and bacteriophages?
There are. There are things called cosmids, for example. They are hybrid vectors
Hybrid vectors.
that combine features of both plasmids and bacteriophages.
So they're like the best of both worlds.
They can carry larger DNA inserts than plasmids. They're easier to work with than bacteriophages.
Wow, that's cool.
And what about those other techniques we talked about, like microinjection and biolistics?
Yeah, when are those used?
Microinjection is often used for animal cells. It's a very precise technique.
Okay.
It allows you to deliver that DNA directly into the cell's nucleus.
So it's very, like, targeted.
Very targeted, like a surgeon
Yeah.
performing a delicate operation. And biolistics, the gene gun method, is often used for plant cells.
Okay.
It's less precise than microinjection,
Yeah.
but it can be very effective for delivering DNA into those cells that have tough cell walls, like plant cells.
So it's like a shotgun approach.
It is.
Okay, that makes sense. It sounds like you need the right tools and techniques
you do.
for the job.
Yeah, it's all about picking the right one.
And it's amazing how much this field has grown.
I know, it's mind-blowing.
What started with those restriction enzymes?
Yeah, just this one discovery.
It's incredible how far it's come.
The future of biotechnology is incredibly exciting, I think,
It is, yeah.
with things like gene editing, CRISPR-Cas9, all these advancements, and our understanding of the human genome is constantly growing.
It's like we're moving into an era where we can not just read the book of life.
We can.
Right, we can edit it.
We can edit it.
That's powerful.
It is.
And
with that power comes responsibility.
yeah, a lot of responsibility.
Yeah, it's important we consider the ethical implications and ensure these technologies are being used for good.
Yeah, very important.
It's been a pleasure discussing all of this with you.
Yeah, this has been
It's a fascinating field.
such an interesting.
I think so, conversation.
And to all our listeners, thank you for joining us on this exploration.
Of the world of biotechnology.
Yes,
It's been a wild ride.
it has.
Until next time, keep those brains buzzing.
Yeah, it really does make you think about like what the future
It does.
holds, you
Yeah.
know? I feel like we just like scratched the surface a little bit
Yeah, just a little bit.
of what all is possible.
Yeah, and that's what makes it so exciting.
Yeah.
It's a field that's always pushing boundaries, challenging how we understand life, and it's opening up these new avenues
Yeah.
for innovation.
It is a little bit overwhelming, to be honest,
It can be,
but in a good way.
in a
Like,
good way.
I feel like I need to go back and reread those textbook excerpts you gave
Yeah.
to really like absorb
Yeah.
all the information.
It is a lot to take in. But I encourage you to do that. I mean, the more we learn about biotechnology, the more we realize how deeply interconnected
Yeah.
it is with our lives. It's
Yeah.
not just about high-tech labs and these complex processes. It's about understanding the very essence of life
Yeah.
and using that knowledge to shape a better future.
Yeah, that's a good way to put it. I think this deep dive has really opened my eyes to the power
It is powerful.
and potential of
Yeah.
biotechnology.
It is an amazing field.
I feel like I've learned a lot, but I also feel a lot more curious about, the world around us.
That's what it's all about, sparking that curiosity
yeah.
and inspiring that exploration. And this is just the beginning of our journey, right?
Right.
There's always more to learn, more to discover, and more to imagine.
Well, to all of our listeners, thanks for joining us on this deep dive into the world of biotechnology. We hope you've learned a lot and that you'll continue to explore this amazing field with us. Until next time, stay curious.